Hey guys, so I promised you guys that we would be going over our FRQs after they're submitted. So what I'm going to do, I'm going to go ahead and record a video going over your first FRQ, so the one for biotechnology and the COMET assay. So we're going to spend some time reviewing that FRQ to make sure that you understand why the answers are the way they're supposed to be. All right, so if I go ahead and switch to the FRQ. Okay, so for this FRQ, you can see that we have what's called a COMET assay. So COMET assays we haven't talked about in class, but that's kind of to be expected with the AP Bio questions. Most of the time they're going to give you some sort of experiment or talk about some sort of analysis that we haven't talked about, but you should have enough basic information to be able to answer this. So if we read our prompt, a COMET assay is a technique used to determine the amount of double-strand breaks in DNA, also known as DNA damage, in cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to the gel that causes the DNA to move, which is electrophoresis, and the DNA is stained with a fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears as a round shape, also known as the head, and the fragments of damaged DNA extend out from the head, which is the tail. The length of the tail corresponds to the amount of damage in the DNA. So if we look at figure 1, we could see right here in this box, there's our head, and then that's what the tail looks like. So we've done gel electrophoresis. We did that in class with the Huntington's disease lab, and so we inserted DNA into a gel, and then we applied an electric current to watch the DNA move. So if I look at question A, so question A says to explain the movement of DNA fragments in the COMET assay, identify one property of DNA and provide reasoning to support how the property contributes to the movement during the COMET assay technique. Well, if we think about DNA, this isn't asking property of DNA from this experiment. It's talking about property of DNA in general. It wants to know, how does electrophoresis work and why? And so if we're talking about our properties of DNA, there's two that we can write about. So our first one is going to be our negative charge of DNA. And the reason why it moves across the gel is because it's attracted to the positive end at the end of the gel. Okay, so that's one property of DNA and why this allows the COMET assay to work, why it allows gel electrophoresis to work. So our other part for our DNA properties, so I don't know if you guys remembered, when we talked about gel electrophoresis, we talked about the DNA moving across in different bands. And so what I'll do is I'll also post up a picture with this about, with gel electrophoresis so you can look at those bands. So if you remember with those bands are the different parts of DNA that are cut up. And so because the DNA has been cut up with the use of restriction enzymes, your DNA is going to travel at different speeds. And so because of that, DNA strands, oops, I can't type, strands have various weights according to size. And so if you remember, the smaller pieces move faster, bigger pieces, wow, I cannot spell today, move slower. Okay, so that's going to be our second property of DNA and the reason why gel electrophoresis works the way it does. So for part B, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Predict the likely results of a COMET assay for this treatment. All right, so some of y'all got this. Some of y'all were a little bit confused, and that's fine. So if we're looking at a COMET assay, the reason why it works is because COMET assays detect double-stranded breaks, and that's what it says right here. They detect double-stranded breaks in DNA. So in this case, this mutagen causes nucleotide substitutions in DNA. So what that means is that if you have, let's say, ATC, maybe you're going to have a GC. So it'll substitute a T for a G. That doesn't mean that you're going to have a break in your DNA. All it's doing is substituting those nucleotides. And so because of that, because you're not causing any double-stranded breaks, more than likely, if the DNA was already intact, you're not going to see any tails. Okay, so no tails will be present with this mutagen. Okay, so you could say no tails will be present, tails will be shorter, okay, heads only, okay, any of those things that indicates that you're not getting double-stranded breaks. You're only having nucleotide substitutions. All right, so let's look up a picture really quick of a gel electrophoresis. Okay, gel electrophoresis. So if I look at an image here, okay, so remember your gel electrophoresis. Here's a good one right here. Okay, so on Wikimedia. So a gel electrophoresis. So if we put in our samples up here at our wells that are at the top of our gel, and then we apply an electric current that's going down, so they're going to be attracted. DNA has a negative charge, so it's going to be attracted down here where there's a positive charge. And so you can see that we have multiple bands for this DNA, and that's because when we applied restriction enzymes, it cut that DNA up into smaller pieces. And so the lighter pieces, the ones that don't have as many nitrogenous bases, are going to be able to move faster than these bigger, heavier ones. All right, so I hope that helps. Let me know if you have any other questions, and I'll post up some other videos to review your other FRQs. Bye.